Imagine standing in a crowd of thousands, seemingly indistinguishable from the sea of faces surrounding you. Yet beneath your skin, inside the nucleus of every one of your trillions of cells, lies a code, a molecular signature so uniquely yours that it has never existed before and will never exist again. But what if that signature contains something extraordinary? What if, encoded within your DNA, are subtle signals of genetic superiority that have shaped your life in ways you have never noticed? Consider this: two identical twins raised in the same household, given the same opportunities, eating the same foods. Yet one excels physically, mentally outlasts their peers, and rarely falls ill, while the other struggles. Scientists have long attributed such differences to environmental factors or random chance. But what if there is something more fundamental at work? What if certain individuals possess genetic variations that provide them with inherent advantages? Microscopic differences that manifest as superior traits in a world that increasingly rewards genetic excellence. You may have dismissed your exceptional memory, unusual resistance to fatigue, or remarkable pain tolerance as normal, perhaps just good luck or the result of hard work. But what if these traits are actually signals of genetic variations that set you apart? What if the code written in your cells contains ancient adaptations and rare mutations that have silently influenced every aspect of your existence? This is not about the superficial genetic differences that create our visible diversity. This is about the hidden architecture of our genome, the 99.9% of DNA that appears identical across all humans, yet contains subtle variations with profound implications. These variations could be the difference between extraordinary and ordinary, between thriving and merely surviving in our increasingly complex world. The truth about human genetic potential has remained largely unexplored, not just because of technological limitations, but because we have been asking the wrong questions. Instead of asking, are some people genetically superior? Perhaps we should be asking, what hidden genetic advantages might each of us possess, and how can we recognize them? Today, we venture beyond the boundaries of conventional genetic understanding to explore the signals of superior DNA that may be hiding within you, clues that have been overlooked by traditional science, but are now being revealed through cutting-edge research and revolutionary insights into the human genome. The story of superior DNA begins not in a laboratory, but in the distant past, approximately 200,000 years ago when anatomically modern humans first appeared. Dr. Julia Quinton, a leading paleogeneticist at the Cambridge Institute for Evolutionary Studies, has spent her career studying what she calls adaptive acceleration in human populations. We tend to think of evolution as an excruciatingly slow process. Dr. Quinton explains, gesturing toward a wall-sized phylogenetic tree in her office. But humans have experienced unusual bursts of accelerated genetic adaptation, particularly in response to extreme environmental pressures. These adaptive bursts have created significant genetic variations that persist in modern populations, variations that can confer remarkable advantages. The conventional understanding of genetics that most of us learned in school, that we inherit half our DNA from each parent through a random assortment of genes, is correct but incomplete. The reality is far more complex and fascinating. Our genetic inheritance is not simply a matter of dominant and recessive traits or predictable patterns of inheritance. It is more like a cosmic lottery where the tickets are not distributed equally, and some combinations unlock extraordinary potential. Consider the case of Jared Quinn, a 32-year-old software developer from Seattle who volunteered for a longitudinal genetic study in 2018. Quinn had always noticed he required less sleep than others, typically functioning well on just 4 to 5 hours per night. He assumed this was simply a personal quirk until genetic testing revealed he carried a rare mutation in the December 2nd gene, present in less than 1% of the population. This mutation allows carriers to enter deeper, more efficient sleep cycles, effectively compressing 8 hours of restorative sleep into less than 5. "I never thought much about it," Quinn remarked when researchers informed him of his genetic rarity. "I just assumed everyone else was exaggerating how much sleep they needed. It was not until the study that I realized I had essentially been gifted an extra 3 hours every day of my life." This December 2nd mutation represents just one of hundreds of potentially advantageous genetic variations that scientists have identified. Others include enhanced oxygen-carrying capacity in blood, superior muscle fiber composition, heightened sensory perception, and even increased cognitive processing efficiency. These are not supernatural abilities but natural variations within our species' genetic diversity, variations that provide measurable advantages in specific contexts. What makes these variations particularly interesting, notes Dr. Olivia Westfield, director of the Human Adaptive Genomics Project at Stanford, is that they often come with no visible external markers. You cannot look at someone and determine if they possess the ACTN3 gene variant that enhances fast-twitch muscle performance or the BDNF-Val66-MET polymorphism associated with enhanced memory formation and protection against stress-induced memory impairment. This invisibility is precisely why so many people go through life unaware of their genetic advantages. Unlike height or eye color, most superior genetic traits manifest in ways that are subjective or contextual. A person might attribute their remarkable endurance to training rather than their rare EPAS1 gene variant that enhances oxygen utilization, the same variant found at high frequencies in Tibetan populations adapted to high-altitude environments. But how do these superior genetic variants arise in the first place? The answer takes us back to the fundamental mechanisms of evolution. Human populations that migrated out of Africa approximately 70,000 years ago encountered dramatically different environments, from the frozen landscapes of northern Europe to the oxygen-depleted elevations of the Tibetan plateau. These environmental pressures became powerful selective forces, favoring individuals with genetic mutations that enhanced survival under specific conditions. Natural selection works like a filter, explains Dr. Marcus Tanner, evolutionary biologist at Oxford University. Beneficial mutations, even small ones that provide just a slight advantage, become more common in a population over time because carriers of these mutations produce more offspring who survive to reproductive age. Over generations, these beneficial mutations can become quite common in populations where they provide an advantage. This explains why certain advantageous genetic variants appear more frequently in specific geographic populations. The ability to digest lactose into adulthood, for instance, evolved independently in European and some African populations where dairy farming emerged, a classic example of convergent evolution driven by similar dietary pressures in different regions. But here is where the story takes an unexpected turn. In our modern interconnected world, where people from diverse genetic backgrounds intermix more than ever before, we are witnessing unprecedented genetic combinations. The child of a Sherpa with enhanced oxygen-carrying capacity and a Scandinavian with superior cold adaptation might inherit both traits, creating a unique genetic profile with advantages neither parent population possessed individually. We are living in the greatest genetic mixing experiment in human history, Dr. Westfield observes. Population groups that evolved in isolation for thousands of years are now combining their genetic advantages in ways that could not have occurred naturally before global travel became commonplace. The genetic diversity within a single modern family can sometimes exceed what existed in entire isolated populations just a few centuries ago. This genetic recombination is not creating superhumans, but it is certainly creating individuals with unique combinations of advantageous traits that may be particularly well suited to our modern environment, an environment that differs dramatically from the conditions under which humans evolved. The implications of this genetic lottery extend far beyond theoretical interest. Your personal genetic profile might contain combinations of variants that influence everything from your disease susceptibility and athletic potential to your cognitive strengths and emotional resilience. And while science has long focused on identifying genetic disorders and risks, we are only beginning to understand the full spectrum of genetic advantages that might be hidden within each of us. As we venture further into this exploration, the question becomes not just whether you might possess superior genetic traits, but how to recognize the signs that have been hiding in plain sight throughout your life. Most people who carry superior genetic variants remain completely unaware of their biological advantages. After all, we have no baseline for comparison. We experience life only through our own genetic lens. How would you know if your cognitive processing speed is exceptional if you have never experienced thinking through anyone else's brain? How would you recognize that your immune system is remarkably efficient if you have never inhabited another body? Despite these limitations, there are observable patterns and experiences that might indicate the presence of advantageous genetic variations. These signals are subtle but recognizable once you know what to look for. Dr. James Richardson of the Genomic Performance Institute has identified what he calls the 7 signals of potential genetic advantage, patterns that often indicate the presence of beneficial genetic variations. The first and perhaps most obvious signal is family history of exceptional longevity or health. Dr. Richardson explains, if you come from a family where multiple members routinely live into their 90s with good cognitive function and minimal health issues, there is a strong possibility you have inherited genetic factors that promote longevity and disease resistance. Indeed, studies of centenarians, people who live to 100 or beyond, have identified specific genetic variants associated with exceptional aging, including mutations in the CETP gene that influence cholesterol metabolism and the FOXO3A gene that regulates cellular stress responses. These genetic factors appear to protect against age-related diseases and promote what researchers call compression of morbidity, remaining healthy until very late in life, then experiencing rapid decline only at the very end. The second signal involves unusual recovery capacity, continues Dr. Richardson. People with certain genetic advantages often report recovering from illness, injury, or exertion noticeably faster than peers. This rapid recovery isn't just a matter of perception. It reflects actual biological differences in cellular repair mechanisms, inflammatory responses, and energy metabolism. Olympic swimmer Michael Phelps provides a well-documented example of this phenomenon. His coaches noted his remarkably rapid recovery between training sessions, allowing him to maintain higher training volumes than competitors. Later analysis revealed that Phelps produces approximately half the lactic acid of typical athletes during intense exercise, a metabolic advantage that significantly reduces recovery time. The third signal involves atypical responses to medications or substances. Dr. Richardson notes, some individuals require unusually high doses of anesthetics or pain medications to achieve standard effects. Others find that caffeine or other stimulants have minimal impact on them. These atypical responses often reflect variations in genes that regulate drug metabolism or neuroreceptor sensitivity. This phenomenon explains why some people can drink coffee before bed with no effect on sleep, while others feel jittery after half a cup. Variations in the CYP1A2 gene, which produces an enzyme responsible for metabolizing caffeine, create dramatic differences in how quickly individuals process caffeine, with some rapid metabolizers clearing caffeine from their bloodstream up to 4 times faster than slow metabolizers. The fourth signal involves environmental adaptability, unusual tolerance for extreme conditions, continues Dr. Richardson. Some individuals demonstrate exceptional capacity to adapt to high altitudes, extreme temperatures, or sleep disruption. These adaptabilities often reflect genetic variations that influence oxygen transport, temperature regulation, or circadian rhythm management. Dean Karnazes, the ultramarathon runner known for running 350 miles without stopping, demonstrates this environmental adaptability. Research has shown that his body produces minimal lactic acid during extended exercise and has an enhanced ability to metabolize fat as fuel. Genetic adaptations that allow him to run continuously for durations that would be impossible for most humans. The fifth signal involves sensory differences, heightened or specialized perceptual abilities. Dr. Richardson explains, this might manifest as exceptional visual acuity, unusual color discrimination, heightened taste sensitivity, or extraordinary hearing discrimination. These sensory advantages often reflect variations in genes that regulate sensory receptor development or neural processing. Consider the case of tetrachromats, individuals who possess 4 types of cone cells in their retinas rather than the typical 3. This genetic variation, found almost exclusively in women, enables perception of an estimated 100 million colors compared to the roughly 1 million colors visible to those with standard vision. Most tetrachromats never realize their perception differs from others. They simply assume everyone sees the world with the same extraordinary color richness they experience. The sixth signal involves cognitive specialization, unusual mental abilities in specific domains, notes Dr. Richardson. This might include exceptional spatial reasoning, mathematical intuition, language acquisition facility, or memory capabilities. These cognitive advantages often reflect genetic variations that influence neural development, neurotransmitter regulation, or brain connectivity patterns. Magnus Carlsen, the chess grandmaster who can simultaneously play against multiple opponents while blindfolded, demonstrates this kind of cognitive specialization. Brain imaging studies have shown that chess experts like Carlsen process chess positions using different neural networks than novices, activating regions associated with pattern recognition rather than analytical processing. This difference appears to have both genetic and developmental components. The final signal involves emotional resilience, unusual capacity to maintain psychological equilibrium under stress. Dr. Richardson concludes, some individuals demonstrate remarkable ability to process trauma, manage anxiety, or maintain motivation through adversity. These resilience factors often reflect genetic variations that influence stress hormone regulation, neuroplasticity, or reward pathway sensitivity. Research on survivors of extreme trauma has identified specific genetic variations associated with psychological resilience, including polymorphisms in genes that regulate serotonin transport, 5-HTT-LPR, and stress hormone receptors, FKBP5. These variations appear to influence how the brain processes and adapts to traumatic experiences, potentially explaining why some individuals demonstrate extraordinary resilience in the face of devastating circumstances. While the presence of one or more of these signals does not guarantee the presence of superior genetic variants, they often serve as valuable indicators. More importantly, they help us recognize that genetic advantages manifest in diverse ways, not just through obvious physical traits or conventional measures of intelligence. The most interesting aspect of genetic advantage is its context dependency, observes Dr. Natalie Chun, behavioral geneticist at UCLA. A genetic variant that provides advantage in one environment might be neutral or even disadvantageous in another. The EPAS1 altitude adaptation gene that benefits Tibetans at high elevations provides no particular advantage at sea level. The point isn't that some people are genetically superior in any absolute sense. It's that each of us might carry genetic adaptations that provide advantages in specific contexts. This context dependency explains why genetic advantages often go unrecognized. If you never attempt endurance sports, you might never discover your exceptional oxygen utilization capacity. If you never experience significant trauma, you might never realize your unusual psychological resilience. Our genetic advantages remain hidden until life circumstances call them into action. But beyond these observable signals lies a deeper layer of genetic advantage, one that operates at the cellular and molecular level, invisible to casual observation but profoundly influential in determining health, longevity, and cognitive capacity. Beneath the observable traits and abilities that might signal genetic advantage lies a deeper level of biological function, the cellular and molecular mechanisms that determine how efficiently your body operates at its most fundamental level. These invisible advantages might never manifest in obvious ways yet provide profound benefits throughout life. One of the most significant of these invisible advantages involves mitochondrial efficiency. Mitochondria, the power plants within our cells that convert nutrients into usable energy, demonstrate remarkable variation in efficiency between individuals. This variation stems partly from mitochondrial DNA, mtDNA, which is inherited exclusively from our mothers, and evolves more rapidly than nuclear DNA. Mitochondrial efficiency isn't something you would notice in daily life, explains Dr. Elena Petrov, bioenergetics researcher at Johns Hopkins University. But it influences virtually every aspect of physical and cognitive performance. Individuals with highly efficient mitochondria essentially get more cellular energy from the same nutrients, which can translate into everything from enhanced endurance to improved cognitive stamina, and potentially even longevity. Studies of elite endurance athletes have identified specific mitochondrial haplogroups, groups of related mitochondrial genetic variations, that appear more frequently in these populations than in the general public. These mitochondrial variants appear to enhance cellular energy production while generating fewer damaging free radicals, a metabolic advantage that becomes particularly valuable during extended physical exertion. But mitochondrial efficiency represents just one category of invisible genetic advantage. Equally important is cellular maintenance, the complex machinery that repairs DNA damage, recycles damaged proteins, and eliminates cellular waste. Every day, each cell in your body experiences thousands of DNA damage events, notes Dr. Robert Jensen, geneticists specializing in DNA repair mechanisms. Cosmic radiation, environmental toxins, and even normal metabolic processes continuously damage our genetic material. What prevents this damage from accumulating and causing cancer or accelerated aging is an elaborate system of DNA repair mechanisms. These repair mechanisms demonstrate significant variation in efficiency between individuals. Some people possess genetic variants that enhance the accuracy or speed of DNA repair, providing substantial protection against cancer and potentially slowing certain aspects of aging. For example, variants in the PRP1 gene, which is involved in detecting and signaling DNA damage, have been associated with enhanced DNA repair capacity and reduced cancer susceptibility. The remarkable thing about superior DNA repair capacity is that you might never know you have it. Dr. Jensen continues. It's not something you feel or experience directly. You might simply be the person who doesn't develop skin cancer despite similar sun exposure as your friends, or who maintains cognitive clarity into advanced age while peers experience decline. Related to DNA repair is cellular stress resistance, the ability of cells to maintain function under adverse conditions such as oxidative stress, temperature fluctuations, or nutrient scarcity. This stress resistance is partly determined by genetic variations in heat shock proteins and other cellular protection mechanisms. Some individuals possess genetic variants that enhance cellular stress resistance across multiple systems, explains Dr. Samantha Torres, researcher in cellular stress biology. Their cells essentially have superior protection mechanisms, allowing them to maintain function under conditions that would impair or damage the cells of most people. This translates into everything from better heat tolerance to enhanced protection against neurodegenerative diseases. Studies of long-lived individuals have identified genetic variations associated with enhanced cellular stress resistance, including variants in the FOXC03A gene mentioned earlier. These genetic factors appear to protect cells against various forms of damage, potentially explaining why some people maintain robust health well into their 9th and 10th decades of life. Another invisible genetic advantage involves inflammatory regulation, the precision with which the immune system deploys inflammation. While inflammation is a necessary defensive mechanism, excessive or prolonged inflammation contributes to numerous diseases and accelerates aging. Some individuals possess genetic variations that optimize inflammatory responses, notes Dr. William Chen, immunologist at Mount Sinai School of Medicine. Their immune systems deploy inflammation precisely when needed, at appropriate intensity, and, crucially, resolve inflammation efficiently once the threat has passed. This optimized inflammatory regulation provides protection against everything from heart disease and diabetes to depression and neurodegenerative conditions. Genetic variations in the IL-10 gene, which produces an anti-inflammatory cytokine, have been associated with reduced inflammatory responses and decreased susceptibility to certain inflammatory diseases. Similarly, variations in the TNF gene, which regulates a key pro-inflammatory cytokine, influence how intensely the body responds to infection or injury. The benefits of optimized inflammatory regulation are profound but often invisible. Dr. Chun continues. You might simply be the person who rarely gets sick, recovers quickly when you do, and doesn't develop the chronic inflammatory conditions that affect so many people with age. But you wouldn't necessarily attribute these experiences to genetic advantage. You might simply consider yourself generally healthy without recognizing the underlying biological cause. Beyond these cellular mechanisms, another category of invisible genetic advantage involves neural efficiency, how effectively the brain processes information, forms connections, and maintains itself. Variations in genes that regulate neurotransmitter production, receptor sensitivity, and synaptic plasticity create significant differences in cognitive function that extend far beyond conventional measures of intelligence. Neural efficiency isn't just about processing speed or memory capacity, explains Dr. Nathan Parker, cognitive neuroscientist. It encompasses how efficiently neural networks form, how effectively the brain prunes unnecessary connections, and how resilient neural systems are to stress or damage. Some individuals possess genetic variations that optimize these processes, providing cognitive advantages that standardized tests rarely capture. For example, variations in the BDNF gene mentioned earlier influence how readily the brain forms new neural connections and how effectively it maintains existing ones. Carriers of certain BDNF variants demonstrate enhanced neuroplasticity, the ability to adapt neural circuits in response to new information or experiences. This enhanced plasticity can manifest as superior learning capacity, particularly for complex skills that require extensive neural reorganization. What's fascinating about neural efficiency is that it often manifests not as genius-level ability in specific domains, but as unusual cognitive resilience and adaptability. Dr. Parker continues. The person with genetically enhanced neural efficiency might not be the one who scores highest on an IQ test, but rather the one who maintains cognitive function despite sleep deprivation, masters new skills with unusual speed, or preserves mental clarity despite brain injuries that would significantly impair others. Studies of individuals who maintain exceptional cognitive function into very advanced age, sometimes called superagers, have identified genetic factors associated with neural resilience, including variations in genes that regulate inflammation in the brain and those involved in clearing protein aggregates associated with neurodegenerative diseases. These invisible genetic advantages, mitochondrial efficiency, enhanced DNA repair, cellular stress resistance, optimized inflammatory regulation, and neural efficiency, rarely announce themselves through obvious traits or abilities, yet they influence every aspect of health, performance, and longevity in ways that become increasingly significant with age. The truly superior genetic variants aren't necessarily those that provide obvious advantages in youth, observes Dr. Jessica Marin, gerontologist specializing in the genetics of longevity. They're often the ones that protect against age-related decline, allowing some individuals to maintain physical and cognitive function decades longer than their peers. These advantages compound over time, becoming most evident in the final third of life. This perspective helps explain why genetic advantages often go unrecognized. Many of the most significant beneficial genetic variations don't provide dramatic advantages in youth, but rather prevent or delay the declines that others experience with age. The true value of superior DNA often reveals itself not in extraordinary achievement, but in extraordinary preservation of function through time. As we continue our exploration, we turn to perhaps the most fascinating aspect of genetic advantage, the emerging science of genetic interaction, where combinations of variants create effects greater than the sum of their parts. The traditional approach to genetic research has focused primarily on identifying single genes associated with specific traits or diseases. This reductionist approach, studying genes in isolation, has yielded valuable insights but fails to capture the complex reality of how our genome actually functions. In truth, genes operate not as isolated actors, but as components of an intricate network where interactions between multiple genetic variants often prove more important than the effects of any single gene. The future of genetic research lies in understanding interaction effects, asserts Dr. Olivia Westfield, whom we met earlier. We're discovering that combinations of genetic variants often produce effects that cannot be predicted by studying each variant individually. It's like a symphony where the music emerges not from individual instruments, but from their harmonious integration. This principle of genetic interaction helps explain why genome-wide association studies, GWAS, often account for only a small percentage of the heritability of complex traits like intelligence or longevity. The missing heritability, the portion of trait variation that should be genetic based on twin studies but cannot be explained by identified genes, likely resides in these complex interaction effects. Consider the case of exceptional longevity, living to 100 or beyond, which demonstrates approximately 25% heritability in twin studies. Researchers initially hoped to identify longevity genes that would explain this heritability. However, extensive studies have found that centenarians rarely possess any single genetic variant strongly associated with extended lifespan. Instead, they typically carry unique combinations of variants, some protective, some neutral, some potentially harmful when isolated but beneficial in specific combinations with other variants. What makes the genetic profile of centenarians remarkable isn't the presence of rare beneficial mutations, explains Dr. Thomas Pearls, director of the New England Centenarian Study. It's the distinctive pattern of interaction between hundreds or thousands of common genetic variants, a pattern that collectively enhances cellular resilience, optimizes metabolic function, and moderates inflammatory responses in ways that promote exceptional longevity. This insight that genetic superiority often emerges from the pattern of interaction rather than from individual mutations has profound implications for how we understand genetic advantage. It suggests that true genetic superiority may not be about possessing specific, superior genes, but rather about carrying combinations of variants that work together with unusual harmony. The science of genetic interaction has been dramatically advanced by two recent technological developments: multi-omic analysis and machine learning approaches to genetic data. Multi-omic analysis integrates information from the genome, DNA sequence, transcriptome, gene expression, proteome, protein production, and metabolome, metabolic products, to understand how genetic variations manifest through these various layers of biological function. Multi-omic analysis allows us to trace the cascade from genetic variation to actual biological function, explains Dr. Michael Zhang, computational biologist at the Broad Institute. We can see not just what genetic variants someone carries, but how those variants influence gene expression, protein production, and ultimately metabolic function. This comprehensive view reveals interaction effects that would be invisible if we looked at DNA sequence alone. Machine learning approaches, meanwhile, can identify complex patterns in genetic data that would be impossible to detect through traditional statistical methods. By analyzing thousands of genetic variants simultaneously, along with environmental factors and lifestyle variables, these algorithms can identify combinations associated with exceptional traits even when no single variant shows a significant effect. The human mind simply cannot process the complexity of interactions between thousands of genetic variants, notes Dr. Zhang. Machine learning algorithms can identify patterns in this high-dimensional data that reveal how combinations of variants collectively influence complex traits like cognitive function, disease resistance, or athletic performance. This research has revealed that some individuals possess what geneticists now call harmonic genomes, genetic profiles where variants across multiple systems work together with unusual synergy. These harmonic genetic patterns often provide advantages greater than would be predicted by the sum of individual variants. Imagine one person who carries 20 genetic variants, each with a small positive effect on cognitive function, explains Dr. Parker. Now imagine another person who carries just 10 such variants, but in a pattern where they amplify each other's effects through network interactions. The second person might demonstrate greater cognitive advantages despite carrying fewer beneficial variants, simply because their particular genetic pattern creates more powerful interaction effects. This concept of genetic harmony helps explain why some people demonstrate exceptional capabilities despite not carrying rare mutations traditionally associated with superior function. Their advantage lies not in possessing unusual genes, but in carrying common variants in combinations that create uncommonly powerful synergies. Evidence for these synergistic effects comes from studies comparing the predictive power of polygenic scores, numerical assessments of genetic predisposition based on thousands of variants, to actual outcomes. Researchers have found that some individuals consistently outperform their polygenic predictions, suggesting their particular pattern of genetic interaction produces effects beyond what would be expected from simply adding up the effects of individual variants. We call these people genetic overperformers, explains Dr. Westfield. Their actual capabilities exceed what their genetic profile would predict based on traditional additive models. This overperformance suggests they possess genetic combinations with powerful synergistic effects, interactions that amplify beneficial impacts while potentially suppressing harmful ones. The phenomenon of genetic overperformance appears particularly evident in studies of cognitive resilience, the ability to maintain cognitive function despite brain pathology or injury. Some individuals with extensive Alzheimer's pathology in their brains nonetheless demonstrate minimal cognitive impairment, suggesting they possess genetic combinations that provide exceptional neural resilience. What's particularly fascinating about these resilient individuals is that they rarely carry any single protective mutation, notes Dr. Parker. Instead, they typically possess unique combinations of common variants that collectively enhance neural protection, promote efficient neural repair, and optimize cognitive network function. It's the pattern of interaction, not any individual gene, that provides their extraordinary resilience. This perspective on genetic interaction offers a more nuanced understanding of genetic superiority, one that recognizes the complexity of how genetic advantages actually manifest. Rather than focusing on isolated, superior genes, it suggests we should consider the harmonic integration of genetic variants across multiple biological systems. The implications extend beyond academic interest. Understanding genetic interaction could eventually enable personalized approaches to education, nutrition, and medical treatment based not on isolated genetic markers, but on comprehensive analysis of how an individual's unique genetic pattern influences their response to different interventions. The future of precision medicine lies in understanding these interaction effects, asserts Dr. Zhang. Two people with the same disease might respond differently to the same treatment because of differences in how their genetic profiles interact with the medication. By analyzing these interaction patterns, we could potentially predict which treatments will work best for each individual based on their unique genetic architecture. This emerging understanding of genetic interaction also helps explain why simplistic approaches to genetic enhancement, such as targeting single genes for modification, often prove disappointing. The complex network of genetic interactions means that changing one component can have unpredictable effects on the entire system, sometimes creating more problems than benefits. Nature has spent millions of years optimizing these genetic networks, observes Dr. Westfield. The superior genetic profiles we observe in exceptional individuals didn't emerge from single mutations, but from the gradual accumulation of variants that work together harmoniously. This harmony cannot be easily engineered through simplistic interventions targeting isolated genes. As we continue to unravel the complexity of genetic interaction, the very concept of genetic superiority evolves. We begin to recognize that true genetic advantage lies not in possessing specific, superior genes, but in carrying a genetic symphony where variants across multiple systems work together with remarkable harmony, creating capabilities and resilience greater than the sum of their parts. This insight leads us to a critical question. If genetic advantage emerges from complex interaction patterns rather than isolated mutations, how do environmental factors influence the expression of these genetic potentials? To answer this, we must explore the fascinating frontier where genes meet environment, the science of epigenetics and gene-environment interaction. If the previous section expanded our understanding of genetic advantage by introducing the concept of genetic interaction, this section takes us a step further by exploring how environmental factors influence the expression of those genetic patterns. This is the domain of epigenetics, the study of changes in gene expression that occur without alterations to the underlying DNA sequence. Your DNA sequence remains largely unchanged throughout your lifetime, explains Dr. Rachel Morrison, epigeneticist at the University of California, San Francisco. But which genes are activated when and to what degree is tremendously malleable. Environmental factors, from nutrition and physical activity to stress exposure and even social interactions, can modify gene expression patterns through epigenetic mechanisms. These modifications can dramatically influence how your genetic potential manifests. Epigenetic modifications take several forms, including DNA methylation, the addition of methyl groups to DNA molecules, histone modification, changes to the proteins around which DNA is wound, and non-coding RNA interactions. These modifications serve as a dynamic interface between our fixed genetic code and our changing environment, allowing for rapid adaptation to environmental conditions without requiring changes to the DNA sequence itself. The implications for understanding genetic advantage are profound. Two individuals with identical genetic variants may express those variants differently depending on their environmental exposures, potentially manifesting dramatically different capabilities and health outcomes as a result. This explains why identical twins, despite sharing virtually identical DNA, can develop different traits and health conditions over time, notes Dr. Morrison. As they experience different environments, their epigenetic patterns diverge, leading to different patterns of gene expression despite their genetic similarity. Studies of identical twins raised apart have provided compelling evidence for the power of epigenetic modification. In one notable case, identical twins separated at birth showed a 33-pound difference in body weight as adults, along with significant differences in susceptibility to specific diseases, differences that correlated with distinct epigenetic modification patterns despite their identical DNA sequences. But the relationship between genes and environment is not simply a matter of environment modifying fixed genetic tendencies. Rather, It involves complex bidirectional interactions where genetic variations influence environmental sensitivity, and environmental factors trigger different responses depending on genetic profile. This concept, known as gene-environment interaction, helps explain why the same environmental exposure, whether nutrition, training, education, or stress, often produces dramatically different outcomes in different individuals. Some genetic variants confer environmental sensitivity. They essentially make the body more responsive to both positive and negative environmental influences, explains Dr. Jason Mitchell, researcher in developmental genetics. Other variants confer environmental resilience. They buffer against environmental influences, maintaining stable function regardless of external conditions. This differential environmental sensitivity helps explain puzzling observations about skill development and performance enhancement. Consider two athletes following identical training protocols. One shows dramatic improvement while the other makes minimal progress despite equal effort. This difference may reflect genetic variations that influence how effectively their bodies respond to training stimuli. Research has identified specific genetic variants associated with training response. For example, variations in the ACTN3 gene mentioned earlier not only influence baseline muscle fiber composition but also affect how muscles adapt to different types of training. Individuals with certain ACTN3 variants show enhanced response to power-oriented training, while others demonstrate better results from endurance-focused protocols. This isn't just about some people having better genes for athletic performance, notes Dr. Mitchell. It's about different individuals having different optimal environmental conditions for expressing their genetic potential. The same person who responds poorly to one training approach might show exceptional progress with a different protocol better aligned with their genetic profile. This principle extends far beyond athletics to virtually every domain of human capability. Educational interventions, nutritional protocols, stress management techniques, and cognitive enhancement strategies all show variable effectiveness depending on an individual's genetic profile. We're moving beyond the simplistic nature versus nurture debate toward understanding nature via nurture interactions, explains Dr. Westfield. The question isn't whether genes or environment determine capabilities, but rather how specific environmental conditions interact with specific genetic profiles to express or suppress genetic potential. This perspective offers a more nuanced understanding of genetic advantage, one that recognizes both the importance of inheriting beneficial genetic variations and the critical role of experiencing environments that optimize the expression of those variations. Consider the case of perfect pitch, the rare ability to identify or reproduce musical notes without reference to external standards. Studies have found that perfect pitch demonstrates both strong genetic influence and critical environmental dependence. Individuals appear to require both genetic predisposition and early musical training during a specific developmental window to develop this ability. Those lacking either the genetic foundation or the timely environmental exposure typically cannot develop perfect pitch regardless of later efforts. This pattern, genetic potential requiring specific environmental conditions for expression, appears consistently across domains of exceptional human capability. Mathematical prodigies typically combine genetic predispositions for numerical processing with early environments rich in mathematical stimulation. Elite athletes pair genetic advantages in areas like oxygen utilization or muscle fiber composition with precisely tailored training environments that optimize these natural tendencies. The truly fascinating cases involve individuals who appear genetically ordinary yet achieve extraordinary results through precisely optimized environmental exposure, notes Dr. Mitchell. These individuals may lack obvious genetic advantages, but possess subtle genetic variations that create unusual sensitivity to specific environmental inputs, allowing them to derive exceptional benefits from environments that produce only average results in others. This insight, that some genetic variations enhance responsiveness to positive environmental influences rather than providing direct advantages, offers a more sophisticated understanding of how genetic superiority manifests. The most valuable genetic variations may not be those that directly enhance capabilities, but rather those that maximize responsiveness to capability-enhancing environments. The implications extend beyond understanding exceptional performance to reconsidering how we approach human development broadly. Rather than providing standardized environments and interventions, this perspective suggests the importance of identifying each individual's unique pattern of genetic responsiveness and tailoring environments accordingly. Personalized education, nutrition, and training aren't luxuries. They're necessities if we want to optimize human potential, argues Dr. Westfield. Each person's genetic profile creates a unique response pattern to environmental inputs. By understanding these patterns, we can provide environments that maximize individual development rather than applying one-size-fits-all approaches that serve no one optimally. Recent technological advances are making this personalized approach increasingly feasible. Direct-to-consumer genetic testing companies now offer assessments of genetic factors influencing everything from nutrition and exercise response to learning style and stress resilience. While these tests remain limited in predictive power, they represent early steps toward truly personalized approaches to human development based on genetic response patterns. The future lies not in genetic determinism but in genetic optimization, asserts Dr. Morrison. Rather than viewing genes as setting fixed limits on potential, we're learning to see them as creating unique response patterns that can be leveraged through precisely tailored environments. The question becomes not, what are my genetic limitations? But rather, what environmental conditions will best express my genetic potential? This shift in perspective transforms how we understand genetic advantage. Superior genetic profiles are not simply those containing inherently better genes, but those offering optimal responsiveness to available environmental resources, creating exceptional capabilities through efficient utilization of environmental inputs rather than through inherent superiority. As we move toward the conclusion of our exploration, we consider perhaps the most profound implication of modern genetic understanding, the recognition that genetic superiority is not absolute but contextual, varying across environments, eras, and domains of function. Throughout history, humans have been fascinated by the concept of inherent superiority, the idea that some individuals possess innate advantages that destine them for greatness. This fascination has often manifested in problematic ways, from aristocratic claims of noble blood to the pseudoscientific racism of the eugenics movement. These perspectives shared a common flaw. They viewed genetic advantage as absolute, universal, and deterministic. Modern genetic science offers a fundamentally different paradigm, one that recognizes genetic advantage as contextual, specific, and probabilistic. This new understanding transforms how we conceptualize superior DNA and its implications for human potential. The entire concept of genetic superiority needs reconsideration, argues Dr. Westfield. No genetic profile is universally superior across all environments and for all purposes. Rather, certain genetic variations provide advantages in specific contexts while potentially creating disadvantages in others. Genetic advantage is inherently contextual and domain-specific. This contextuality appears consistently in research on genetic variations associated with enhanced capabilities. The COMT-Val158 MET polymorphism, for instance, influences dopamine regulation in the prefrontal cortex, affecting cognitive function. Individuals with the Val variant demonstrate cognitive stability, maintaining consistent performance under stress, while those with the MET variant show cognitive flexibility, superior performance under optimal conditions but greater vulnerability to stress-induced impairment. Neither COMT variant is inherently superior, explains Dr. Parker. The Val variant provides advantages in high-pressure situations requiring consistent performance, while the MET variant offers benefits in low-stress environments requiring creative problem-solving. Their relative value depends entirely on context. This pattern, genetic variations providing context-dependent advantages rather than universal superiority, appears consistently across domains. The warrior gene variant, MAOAL, associated with increased aggression under provocation creates disadvantages in peaceful contexts, but may provide advantages in conflict situations. The sickle cell trait creates vulnerability to sickle cell anemia but provides protection against malaria, a trade-off that becomes advantageous or disadvantageous depending on malaria exposure. Beyond contextuality, modern genetics reveals that genetic advantage is typically domain-specific rather than general. Most genetic variations influence specific aspects of function rather than creating broad-spectrum superiority. The public often imagines superior genes creating generally enhanced humans, smarter, stronger, healthier across all metrics, notes Dr. Jonathan Reynolds, behavioral geneticist at King's College London. but actual genetic advantages typically influence specific capabilities or traits. The same person might possess genetic variations enhancing cardiovascular efficiency, while carrying others that increase vulnerability to autoimmune conditions. Genetic advantage is rarely universal across domains of function. This domain specificity helps explain why individuals who demonstrate exceptional capabilities in one area often show typical or even below-average function in others. The genetic variations that enhance specialized neural processing for mathematical computation may provide no advantages, and sometimes create disadvantages for social cognition or emotional processing. Perhaps most importantly, modern genetics reveals that genetic advantage is probabilistic rather than deterministic. Genetic variations influence tendencies and potentials rather than determining fixed outcomes. Even the most powerful genetic variants merely shift probabilities, explains Dr. Reynolds. They create predispositions and tendencies, not predetermined outcomes. Environmental factors, random developmental events, and personal choices all interact with genetic tendencies to produce actual capabilities and characteristics. This probabilistic nature of genetic influence appears clearly in studies of genetic variants associated with educational achievement. While dozens of genetic variants have been associated with educational outcomes, even individuals carrying numerous education-positive variants can fail academically in unconducive environments, while those with fewer such variants often thrive when provided with appropriate educational support. Genes are about potentials, not guarantees, asserts Dr. Reynolds. They establish ranges of possibility and patterns of responsiveness rather than fixed destinations. The actual manifestation of genetic potential depends on complex interactions between genetic predispositions, environmental exposures, developmental timing, and individual choices. This new paradigm, recognizing genetic advantage as contextual, domain-specific, and probabilistic, fundamentally transforms how we understand the concept of superior DNA. Rather than viewing genetic superiority as an absolute quality creating universally enhanced humans, we recognize it as a complex pattern of variations that create specific potentials requiring appropriate conditions for expression. The person with truly superior DNA isn't necessarily the one carrying the most obviously advantageous variants, observes Dr. Westfield. It's the person whose specific genetic pattern creates optimal alignment with their particular environment, lifestyle, and pursuits. Genetic superiority is about congruence between genetic tendencies and life circumstances rather than about inherently better genes. This perspective helps explain why many individuals with remarkable genetic potentials never manifest exceptional capabilities, while others with seemingly ordinary genetic profiles achieve extraordinary results. The expression of genetic potential requires alignment between genetic predispositions and environmental conditions, alignment that often occurs through either fortunate circumstance or deliberate optimization. Some people achieve alignment through luck, being born into environments naturally suited to their genetic predispositions, notes Dr. Reynolds. Others achieve it through exploration and self-discovery, finding the domains and approaches that best express their particular genetic tendencies. Still others create alignment through deliberate environmental modification, tailoring their surroundings to optimize the expression of their genetic potential. This emphasis on alignment rather than inherent superiority offers a more empowering perspective on genetic influence. Rather than viewing genes as fixed limitations or predetermined advantages, we can recognize them as creating unique patterns of potential that can be optimally expressed through appropriate environmental conditions and life choices. The science of genetic potential suggests a fundamental shift in how we approach human development, argues Dr. Westfield. Rather than imposing standardized expectations and environments, we should focus on helping each person identify their unique pattern of genetic responsiveness and creating conditions that optimize the expression of their particular potential. This approach, genetic personalization rather than standardization, represents the frontier of applied genetic science. By understanding how specific genetic profiles respond to different environments, interventions, and practices, we can potentially help each person express their genetic potential more fully. The implications extend far beyond individual development to societal design. Our educational systems, workplaces, healthcare approaches, and social structures have traditionally been designed for standardized humans rather than for the genetic diversity that actually exists. By recognizing the variety of genetic response patterns present in human populations, we might design more inclusive systems that accommodate diverse genetic predispositions rather than privileging specific genetic profiles. A truly advanced civilization wouldn't be one that enhances genes to create standardized, superior humans, suggests Dr. Reynolds. It would be one that recognizes genetic diversity as a fundamental resource and creates environments that allow diverse genetic potentials to express themselves optimally. The goal isn't genetic uniformity but genetic optimization, helping each unique genetic profile find its optimal expression. This perspective offers a more sophisticated understanding of what it means to possess superior DNA. The advantage lies not in carrying specific superior genes, but in possessing a genetic profile that creates opportunities for exceptional expression in domains and contexts that matter to the individual and their society. As we conclude our exploration, we return to where we began, considering what it means to recognize the signals of genetic advantage in your own life and how this recognition might influence your approach to developing your potential. We began this journey by asking whether subtle signals in your life might indicate the presence of advantageous genetic variations that provide opportunities for exceptional function in specific domains. Through our exploration, we've discovered that genetic advantage is far more complex, contextual, and personalized than traditional conceptions of good genes suggest. The truly valuable insights come not from searching for evidence of genetic superiority, but from recognizing your unique genetic signature, the distinctive pattern of genetic variations that shapes your responses to different environments, activities, and challenges. This signature creates a personal landscape of genetic potential with distinctive peaks, domains of potential excellence, valleys, areas of potential vulnerability, and response patterns, how you uniquely react to different inputs and interventions. The question isn't whether you have superior DNA in some absolute sense, concludes Dr. Westfield. It's about understanding your genetic uniqueness and its implications for how you might optimally develop your particular potential. Everyone possesses genetic variations that create opportunities for exceptional function in specific contexts. The challenge is identifying and optimizing those opportunities. How might you recognize aspects of your genetic signature without undergoing extensive genetic testing? Several approaches offer insight. First, pattern recognition, identifying consistent themes in your experiences and responses. Do you consistently demonstrate unusual capabilities or limitations in specific domains? Do you respond atypically to certain substances, environments, or interventions? These patterns often reflect underlying genetic tendencies. Pay attention to areas where you seem to differ consistently from others, suggests Dr. Reynolds. If you recover from intense exercise notably faster than training partners, maintain mental clarity with minimal sleep, or learn certain types of information with unusual ease, these differences often reflect genetic variations affecting relevant biological systems. Second, family observation, recognizing shared patterns across genetically related individuals. Do certain unusual capabilities or traits appear repeatedly in your family? These recurring patterns often indicate inherited genetic variations with significant functional effects. Families often share not just specific traits but patterns of potential, notes Dr. Reynolds. You might notice that relatives demonstrate exceptional capabilities in related domains, some artistic, others mathematical, but all involving similar cognitive processes. These patterns often reflect shared genetic variations affecting neural development or function. Third, response mapping, systematically observing how you respond to different environmental inputs and interventions. Do certain dietary patterns, exercise approaches, learning methods, or environments consistently produce better or worse results for you than for others? These differential responses often reflect genetic variations affecting how your body and brain process specific inputs. Self-experimentation can reveal aspects of your genetic signature that might otherwise remain hidden, suggests Dr. Westfield. By systematically varying inputs, whether nutrition, physical activity, learning approaches, or stress management techniques, and carefully observing results, you can identify patterns that reveal your unique genetic response tendencies. Beyond recognition comes the challenge of optimization, creating conditions that best express your genetic potential while mitigating vulnerabilities. This optimization involves both environmental selection, finding contexts that naturally align with your genetic tendencies, and environmental creation, modifying your surroundings to better support your genetic potential. Some genetic variations create narrow bands of optimal function. They provide exceptional capabilities under specific conditions, but create vulnerabilities under others, explains Dr. Morrison. Other variations create broad response patterns. They offer moderate advantages across diverse conditions. Understanding your particular pattern helps determine whether you should seek specialized environments or create adaptable ones. This process of genetic optimization represents perhaps the most profound application of genetic science to human development. Rather than using genetic information to select or modify humans, we can use it to select and modify environments, creating conditions that allow each person's unique genetic potential to express itself optimally. The future of genetic science isn't about designing better humans through genetic modification, asserts Dr. Westfield. It's about designing better environments through genetic understanding, creating conditions that support the optimal expression of the genetic diversity already present in human populations. This perspective transforms how we understand both genetic advantage and human potential. True genetic advantage lies not in possessing universally superior genes, but in achieving optimal alignment between your unique genetic signature and your life circumstances, creating conditions where your particular pattern of genetic potential can express itself fully. The signals of superior DNA are not found in simplistic measures of intelligence, physical prowess, or health outcomes. They appear in the distinctive pattern of capabilities, responses, and potentials that constitute your genetic uniqueness. A pattern that creates opportunities for exceptional function when properly understood and optimally expressed. As our genetic knowledge continues to advance, we move not toward a world of genetically engineered uniformity, but toward one of genetically informed diversity, a world where each unique genetic signature finds conditions that support its optimal expression. This isn't genetic determinism, but genetic empowerment, using genetic understanding not to limit potential but to more fully realize it. Your DNA is neither superior nor inferior in any absolute sense. It is uniquely yours, a distinctive pattern of genetic variations unlike any other in human history. The true advantage lies not in what genes you possess, but in how fully you understand and express the unique potential they create. In this recognition of genetic uniqueness rather than genetic hierarchy, we find a more scientifically accurate and humanely empowering vision of what it means to possess remarkable DNA. The most profound signal of genetic advantage may be this: not the possession of specific superior genes, but the achievement of harmony between your distinctive genetic signature and the life you create. A harmony that allows your unique potential to manifest itself fully in a world that needs the diverse capabilities our genetic diversity makes possible.